All right, this video was created to help you understand cell reproduction. It's going to break it down and hopefully make it more simple. So, cell reproduction, what's going on? That's the big question. So, we want to use cell reproduction depending on which type, but when cells get too old, worn out, or too big, basically they need to be replaced. So, if you look at the diagram on the left, right here, it is showing the life cycle of a cell. It has three parts where you have interphase, which is this large part over here, which takes up most of the cell cycle, including G1, S, and the G2 stage. And then after that, you go through mitosis. And then the very last part over here is cytokinesis. So there's three parts: interphase, mitosis, and cytokinesis. All right, here's just a diagram comparing mitosis and meiosis. Those are the two types of cell reproduction we're going to talk about, mitosis and meiosis. It kind of just gives you a brief overview as far as the starting point and the end result. So in mitosis, you start with a diploid cell, which has 46 chromosomes, and then you're going to end with two identical diploid cells. So they're going to be just like the parent cell, and they're going to be identical, both having 46 chromosomes. In meiosis, you will start with the diploid cell. They will then replicate, and then you will end up, after they replicate again, with haploid cells, 4 haploid cells with 23 chromosomes in it. So again, you can see that the end result is definitely different. There's only one division through mitosis, and you end up with identical cells. And then in meiosis, you have 2 separate divisions, and you end up with 4 haploid cells. All right, just another Venn diagram to help compare and contrast mitosis and meiosis. In mitosis, over here on your left side, the cells that go through mitosis are body cells. Basically, any cell other than the sperm and the egg. So skin cells, liver cells, hair cells, things like that. So body cells go through mitosis because you need an exact copy. They are diploid cells, which means they have 2 copies of the chromosome, of each chromosome, and then they go through 1 cellular division, so they only divide 1 time, and then you end up with 2 identical daughter cells. Over on the meiosis side, you only use meiosis when you're trying to create sex cells, which are, again, for males and humans, sperm, and for females, the egg. You create 4 haploid daughter cells, and you go through cell division twice. And then you end up with 4 daughter cells, 4 haploid daughter cells. The things that are similar is they are both, the chromosomes are replicated in both processes, and they're both types of cell division, and they both occur in phases. All right, cellular reproduction. What are the types? We talked about both of them already, mitosis and meiosis. So here's some information about mitosis. It's a process of the nucleus dividing. So that's what's going on with mitosis. The actual nucleus and everything inside is dividing. There are 4 phases in this process: prophase, then metaphase, anaphase, then telophase. So let's talk about prophase. That's the first phase. Key things to remember is that the chromatin condenses into chromosomes. So in the next screen, you'll see a chromosome, which looks like a small X. Those are the chromosomes. And basically, it's kind of like long, stringy, spaghetti-like things. Or you can think about yarn or string stretched out. And if you coil it up and roll it up and ball it up, then you can see it better. So those are the actual chromosomes coiled up and balled up that you can see now. And the nuclear envelope disappears. Here's second phase, metaphase. These are the chromosomes I was talking about in the last slide. They are the chromosomes that carry our DNA, long story short. So they line up in the middle in metaphase. That's the key things that happen. The chromosomes line up in the middle, and the spindle fibers, these are the spindles, and these are the fibers coming from the spindles. These spindle fibers attach to the chromosomes at the centromere. So they attach in the middle of the chromosome, and the area is called the centromere. The third phase is anaphase. those chromosomes that were lined up in the middle and these spindle fibers attached, once the spindle fibers shorten, that actually separates the chromosomes. So it pulls them to each side of the cell. So you see these now no longer connected and they're being pulled to the other side. That is anaphase. Last phase of mitosis is telophase, and it's when you have basically the nucleus has already divided and been replicated, and you're basically about to separate into a new cell. So it's almost fully divided. You see the cleavage furrow right here, which is like basically just a little indentation because the cells are kind of like pulling apart, about to separate. So it's almost two cells. After mitosis, remember, mitosis is only those 4 steps. So cytokinesis comes after. It is not part of mitosis. But what happens is the cytoplasm, which in this screen right here, this is all cytoplasm. All in between here, it's still connected. The cytoplasm is attached. It needs to be separated. It gets separated in cytokinesis. And then you have 2 fully functional cells. All right, 2 separate cells. All right, now that was mitosis. Now we're going to move on to meiosis and cover it pretty quickly. This diagram shows everything that you really need. You can find it in any biology textbook as well, or online where I found this picture. Meiosis is when you're making sex cells. So it goes through the same process as mitosis, which means prophase, metaphase, anaphase, and telophase, but it goes through it twice because there are 2 cell divisions, and it eventually produces 4 haploid daughter cells. You look at it over here, you kind of have 1 cell, 1 cell, 1 cell. After it goes through those phases, then it divides 1 time here. You have 2 cells, 2 cells, 2 cells, 2 cells, and then it divides here, and you end up with 4 haploid cells. All right. The purpose of going through 2 divisions, hopefully you can kind of see it through this diagram right here. So you start with tetrads, and those tetrads must divide once to get duplicated chromosomes, then divide again to get unduplicated chromosomes. So here would be your tetrad or your duplicated homologous chromosome. Yes, I know it's a lot of words. Duplicated, which means there's copied. So there's a copy here and a copy there, and they are homologous chromosomes. All right, so this thing together would be a tetrad, this whole thing. 1, 2, 3, 4 at the top, and 1, 2, 3, 4 at the bottom. So the first round of division is to separate these 2 things. So right now this is all 1 cell. And then you want to separate it to where you have 2 cells, 1 cell having one of these in it, and another cell having the other one in there. So these are the duplicated chromosomes. And then you need to do the division 1 more time in order to separate it and get 4 separate cells that have just this unreplicated, unduplicated chromosome in it. So the goal is to get this haploid cell, which comes from these. The end of meiosis, we said we have 4 sex cells. Meiosis ends with 4 haploid daughter cells. 1, 2, 3, 4. They are not identical and have genetic variability. Genetic variability, which means they have different combinations of genes. So this cell has certain types of genes in it. This cell has another alternative form of those genes along with this cell and this cell. So example, this one might be, I don't know, might have, they all have hair color in them, right? Hair color would be the gene. This has a brown trait, this has a black trait, this has a blonde trait, this has a red trait. So they're all cells from the same organism, but depending on how it matches up with the egg in the female, they can give you different combinations of those genes. All right, I hope this made mitosis and meiosis a little more clear. You can watch it over and over again or take notes. I hope it helps you. All right, thanks.